Hello, my name is Sергeyeva Жанна, and I have something I want to tell you about plasmids. I would like to tell you just some interesting facts about their diversity in nature and their necessity for bacterial cells. We are going to talk about bacterial plasmids, although sometimes plasmids can be present in the cells of multicellular organisms, but we're going to focus on the plasmids of bacteria. So, bacterial plasmids. What are these things and what are they for? And actually, my PhD thesis were dedicated to the study of cryptic plasmids of phytopathogenic bacteria. Well, if it's not that difficult to understand what does phytopathogenic bacteria means, it means that these bacteria infect plants. Then to understand what is a cryptic plasmid for an average person is rather difficult. And I think that after this short course, you will definitely understand all the basic things about plasmids and their use in biotechnology, the creation of vectors, and all the other interesting things. So let's begin. First of all, every big thing starts with just small steps, small things. And the first thing we need to do is to understand what exactly is a plasmid. Well, we must try to give a definition, but not just give a definition, but also to understand what is it. So let's begin. Well, first of all, plasmids are small circular DNA molecules. They are present within the cell with the chromosome. So the cell contains chromosome and it contains a small plasmid, a big plasmid. Well, plasmid is something that is extra the main chromosome of the cell. Small, stable, and easy to manipulate. That's why they are used in biotechnology. And you can see that this small plasmid is present in the bacteria. We're going to talk about this. Okay, so the definition. Plasmids are extrachromosomal, autonomous, circular genetic elements. And what is it about? Let's begin with the end, genetic elements. There are actually a lot of genetic elements or structures, genetic structures that can be called elements. Usually they are called altogether mobile genetic elements. Plasmids are also mobile genetic elements. There are also different genetic elements like insertional sequences, like transposons, plasmids again, prophages. These are all mobile genetic elements. It means that these are pieces of DNA or RNA molecules, and they can move within the genome of bacterial cell, different cells actually, not only bacterial but also eukaryotic cells. And they can also move between different cells, between organisms. So these elements are called mobile genetic elements. Plasmids are also mobile genetic elements. They are genetic elements. They are presented by DNA molecules. There are some RNA plasmids, but we are not going to focus on them. We're going to talk about DNA plasmids. Genetic elements, circular. That's an easy thing to understand because they are closed in a circle. Although there are some linear plasmids, again, we are not going to talk about them because they're rather rare. We are going to talk about frequent thing, circular plasmids. Autonomous. It means that their life cycle, their replication, the activity of their genes doesn't depend on the chromosome, on the activity of the chromosome. It means that plasmid lives its own life in the cell. It can travel from one cell to another cell. It can replicate and make as many copies of it as it's needed for its properties. Well, anyway, it is autonomous and it's extrachromosomal. It means that it's beyond the chromosome. We have the chromosome in every cell because it's absolutely vital genes that are needed for the survival of this bacterial cell of any organism. And our plasmids, and our plasmids are also considered to be part of the whole cell genome, but they are not part of the chromosome. They are out of it, extrachromosomal. So now you understand the full definition of a plasmid. Plasmids are extrachromosomal, autonomous, circular genetic elements, usually presented by DNA molecules. The amount and copy number. Well, these are actually a little bit different things if we talk about plasmids. Because as you can see on the picture, This bacterial cell contains plasmids. 1, 2, 3, the total amount of the plasmids. But these are different plasmids. One of them is bigger or larger, and 2 are smaller. Actually, if we talk about the copy number, so the larger plasmid is presented in only 1 copy, and the smaller plasmids are presented in 2 copies. But there are only 2 types of plasmids in this bacterial cell. It means that the total number of plasmids means how many types of plasmids the cell contains. And if we talk about copy number, it can be different for every single plasmid. It's like a feature of a plasmid, a characteristic. And that's what we're going to talk about right now. So, first we must focus on the size of a plasmid, different plasmids. Actually, when we start talking about plasmids, we usually talk about small DNA molecules, little plasmids. Well, they are rather small. They are much smaller than the chromosome, although there are some bacteria that contain big plasmids that are just almost as big as the chromosome. And usually, when we try to understand which molecule is a chromosome and which is a megaplasmid, we try to focus on the genes, and we think that vital genes must be on the chromosome, and non-vital genes must be on the plasmid. But sometimes plasmids carry really very important genes, and this characteristic cannot help us. So anyway, it's sometimes really difficult to differ a megaplasmid from a chromosome. What is a megaplasmid? A megaplasmid is a big plasmid. It usually contains a lot of interesting genes that can be used in biotechnology. But small plasmids are better to be used in biotechnology because of their size, because they can easily move between the cells. We can take them out, we can put them in. It's rather difficult to perform this with megaplasmids because they are, because of their size, they can actually be ruined while these manipulations. But they contain very interesting genes. It means that if I look forward, we can take these genes out of megaplasmids and put them into small ones if we want to create a vector. So, one more time. Plasmids can differ in their sizes. Usually, plasmids are rather small molecules, but sometimes there can be megaplasmids in the cell. Those megaplasmids are usually presented in a low copy number, like 1 to 3 copies per cell, but they contain very interesting genes. So these are megaplasmids. Copy number. As I already said, there can be different types of plasmids in the cell, and these different types can be characterized by different copy number. How many copies of this particular plasmid can be present in this bacterial cell? There are plasmids that are characterized by low copy number. It means that there are about 1, 2, maybe 5 copies of this plasmid in the cell. High copy number plasmids can be presented in the cell in hundreds of copies, like 3, 5, 700 copies per cell. Usually, it is true for small plasmids, because megaplasmids are usually present in low copy number. There is also a middle class plasmids that are presented in the cell in 20 to 50 copies, something like this. And what is also very interesting, if the cell contains plasmids, well, it contains one type of a plasmid that is presented in a number of copies, let us say 100 of copies. And these plasmids encodes resistance to some sort of antibiotic. So, if we put this cell in the condition where it is influenced by this antibiotic, on the medium with this antibiotic, the number of copies will increase because of the need of the product product of this plasmid to be for this cell to be resistant to antibiotic. And it means that we will have an increased number of copies. Although there were already a lot of copies of this plasmid in the cell, we can even increase this number by the pressure, specific pressure, if we put this cell on the medium with antibiotic. How can we use it? If we need to isolate the plasmid from the cell, we need as much copies as we can get. So we can present this procedure before the isolation of a plasmid, increase the number of plasmids to get a bigger number of them if we isolate this plasmid. So some plasmids can travel through bacterial cells of different species. They are called broad host range. It means that bacteria of different species can carry one and the same plasmid. That is absolutely normal for this plasmid to travel through species, to be widely presented in nature. But there are also plasmids that are characterized by a narrow host range. It means that we can find these plasmids only in some certain species of bacteria or closely related species. They don't like traveling through different bacteria. They have their own special friends, and they just stay true to this friendship. So these are narrow host range plasmids that are connected with some certain species of bacteria, and broad host range plasmids that can be present in different species of bacteria. How can we use this information? In biotechnology, it's very important, because if we want to create a vector, an instrument to deliver different genes, we need broad host range plasmids, those that will not see borders to how to say it. They don't see borders to replicate and produce some sort of products in different bacterial species, in different types of bacteria. Because if we talk about vectors based on the narrow host range plasmids, they can really be more like effective in only certain species. So our circle of interests becomes narrowed by certain species of bacteria. And with broad host range plasmids, we can use them to many different bacteria. So it's a very nice instrument to change, to genetically modify different bacteria. So broad host range are more preferred, broad host range plasmids are more preferred for the creation of vectors in biotechnology. So if we talk about the spread of plasmids in nature, well, actually, almost 50% of bacteria contain plasmids. One plasmid or two different plasmids in a certain copy number, that is a feature of these plasmids. And these plasmids are like additional genetic luggage. So imagine yourself going to travel. You take a backpack and you put everything you need to this backpack so that your hands are free and your legs feel good and you can travel. And this backpack is like a genome. So all the necessary genes are there. And plasmids are like additional bags that you can carry in your hand, but if you carry them, you get tired and your hands cannot function because they have these bags. Anyway, it's like taking an extra pair of shoes. In case of the rain, you will have dry shoes to put on. But if there will be no rain, you will just carry those boots. And it's like with the plasmids. A bacterium carries the plasmid, and it's like an additional number of genes. They can be useful because they can give bacteria an ability to be resistant to antibiotics, to find new pathways to eat something new, metabolic pathways. It can give a bacteria an ability to produce toxins and cholecines, special molecules that kill other bacteria. So in the competition of survival, sometimes plasmids really give an additional qualities that can help bacteria to survive. But anyway, it's an additional genetical luggage because it takes energy, it takes materials and from the bacterial cell to maintain this plasmid, to maintain the product of this plasmid. So it's like a philosophical choice because the plasmid gives additional qualities, but it also takes energy and time and power from our bacterial cell. But anyway, plasmids are widely spread in nature and presented in different types of bacteria, and plasmids can carry very interesting genes that can be used in biotechnology, and it's plasmids, because they are autonomous, they can replicate by themselves, are really very important models if we can put some new genes inside of this plasmid, and we can, with using this plasmid, we can make this bacterial cell just a new factory for production of new substances. So that's why plasmids are widely used in biotechnology. And these are just basic facts about plasmids, and we're going to continue our conversation about the diversity and necessity of plasmids for bacterial cells. But anyway, as for this one, I think this is it. I think it will be enough information. So thank you for your attention and see you next time. Goodbye.